This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 177, recorded on May 24th, 2018. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How are you? We missed you for a week, right?
Between now and the last time we talked, my son was married in Kalamazoo, so that was a lovely
Congratulations.
event. Yeah.
Hey, mazel tov.
Nice.
And it's golf season here in Michigan, so I'm happy.
That's what you like. Also joining us from Small Things Considered, Elio Schaechter.
Hello there.
Welcome back. I think you were here last time, right? Yeah.
I was here. Yeah, I think so.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
I wasn't too far from you yesterday. I was in Birmingham, Alabama.
That's still pretty far.
In the U.S. scheme of things, it's closer than California, right?
That is indeed true.
Do you remember the New Yorker map of the world?
I was just going to say, you New Yorkers think it's like really a long way.
Yes.
I know
There's
this.
a vast nothing between the Hudson River and the West Coast, right?
That's right. You had New York, the Hudson River, and then California.
Yeah, that's why we refer to them as the flyovers.
Yeah, Central Park is larger than the Midwest.
Yes.
Well, at one time, many years ago, there weren't too many people out west.
That's true.
And there weren't any sports teams. They all were here and then they left.
To go to the West Coast.
Yeah, people started leaving the cities in the Northeast, right? And they moved west. And so everything else went with them. But now, that poster comes from those old days, I think. But yeah, when I travel, I always say, I'm close to South Carolina. Not really, but it's all right. Yes, I was in Birmingham, Alabama.
What was happening there?
I gave a talk at the University of Alabama.
UAB.
UAB.
Yeah, great.
And if you're ever there, go to Lucy's for coffee in the morning. That's the place to go.
Pour overs?
Yeah. And Lucy will make your coffee for you. She's been running the place. She used to have a cart outside on the street, and she made enough money to rent a space, and it's really good. And I got my picture taken with her.
All right. And
she knows all the virologists that have come through UAB because they all get coffee there.
Oh, that's great.
All right. I think this is our last show before ASM Microbe, right?
It is.
Yeah.
Let's see. Today is, almost the end of May. And then next week is the 30th. And that's right. Then the next week, we would normally record on Thursday the 7th, but instead we'll be recording on Sunday, June 10th. And let me tell everyone the time. It is 10:30 a.m. in Atlanta. So we'll be in there live. All of us will be there with a guest, and you should come listen to us.
Say hello.
Not just listen to us. They can even look at us.
They could look at us. And I will bring some T-shirts, some TWiM T-shirts to throw out into the audience.
Wow. I hope I catch one.
But of course, if no one's in the audience, then I'll just give them to Michele and Elio and Michael.
Okay.
Well, I think we have to put in to our esteemed executive director T-shirt cannon into his budget for next year, Michele. As you're going to be the president, you can put in a T-shirt cannon.
I looked into one of those.
You did?
They're not cheap.
No.
And
Bill Murray.
I don't think you could bring them on an airplane.
You probably can't. Bill Murray is one of the co-owners of our minor league baseball team. And the minor league baseball team park is always selling out. And they do all these promotions. And they have the T-shirt cannon going out. And Bill Murray goes out there. Bill Murray from Caddyshack fame and Saturday Night Live. He goes out there and fires the gun. And people clamor for T-shirts. And every Friday night when they're in town, there's fireworks.
I always wanted to fire T-shirts, but I'll just throw them out.
Well, we could just bring a few lacrosse sticks.
Yeah, that's right.
I've seen our lacrosse team here at Michigan has used those to loft T-shirts up into the basketball arena.
That's not a bad idea. Yeah.
Yep.
I have sticks left over from my kids.
All right.
They're sitting in the garage. All right, let's talk about some microbiology because this, after all, is This Week in Microbiology. And today we have a snippet and a paper. We have no email so that we can spend all our time on these papers. And Michael, tell us what you have.
So I picked a paper from the Journal of Applied and Environmental Microbiology, one of the ASM's flagship journals, and it's entitled A Decade of Streptococcus Thermophilus Phage Evolution in an Irish Dairy Plant. And the paper was authored by Lavelle, Murphy, Fitzgerald, Luigi, Zomer, Neve, Ventura, Franz, Chambalou, Cinderin, and Mahoney.
All right, let's Elio and I tell you how to pronounce Lully.
Lully?
Right, Elio?
Lully.
You make
I
it
don't
all-
think my tongue is connected like that.
It's from Parma.
Parma, yeah.
He's from Parma. And the paper is from the University of Cork Island, the School of Microbiology, and Parma, Italy. They're pro-genomics in the Department of Chemistry and Life Sciences, and the Department of Microbiology and Biotechnology from the Max Rubner Institute in Kiel, Germany. And I picked this paper principally because I think most of our audience will appreciate that phages are viruses of bacteria and can result in one or two behaviors to the bacterium, either death as a consequence of lysis as the phage produce their progeny, or integration of the phage genome into the microbe. And often lysogeny can confer behaviors within the host. And in the world of fermented products, either lysis, where the cells are effectively wiping everything out, or the phage are wiping everything out, or lysogeny might result in a bad outcome to the people fermenting the product. And in this case, since we're talking about cheese, they're interacting with milk. And so here the authors were concerned with the phages of Streptococcus thermophiles, as it's one of the most extensively employed commercial starter microbes used widely for the manufacture of these fermented milk products, such as yogurts and various cheese. In this particular strain, or as they abbreviate it throughout their paper as ST, is used to produce a particular Irish hard cheese similar in style and flavor profile to cheddar. And it's at this particular plant that they studied, it's only produced 3 months of the year. And the rest of the time, the plant is producing whey protein. And many of our weightlifting friends out there use whey protein to help put on weight and to bulk up and to do all sorts of other things.
That's what you use to put on muscle, Michael?
No, I just put on weight, it seems. The reason they are concerned is a phage infection of an ST starter culture may result in an incomplete or failed fermentation. And so the cheese may not develop the flavor profile, may develop a different flavor profile, the texture might not be right, and thus it can have considerable economic consequences to the dairy industry.
Michael, when this happens, where does the phage come from?
The phage is literally in the air and it just falls in because they're ubiquitous. And part of this is, I think, and this is one of the reasons I want people to not only look at this paper for the results that they observed, but to use it as a guide to think about all of the stuff that's coming out about the microbiome and some of the important controlling studies that we must do as we begin to take the microbiome apart, design experiments, because a lot of the papers in the microbiome world aren't considering the viruses of the microbiome. And, you know, it's talked about as the virome, but we have not yet seen it. And so I picked this paper principally because it shows you how an industry where the starter culture is important, the behavior of the starter culture is important in terms of creating a product. And in the case of looking at the microbiome and all the other things, we want to think about how we need to design these experiments. So this is a nice, straightforward paper that is rigorously analyzed. And that's the hallmark of this paper. They have 10 years worth of data packed into this paper. And the other reason is once upon a time, I was humbled by a fugitive phage infestation. It was during a phase of my research career where we were using a culture of a methylotrope, Methylobacterium, at a cell density of around an OD of 10. And the methylotrope I was using had this beautiful color. It looked like a strawberry milkshake. And at an OD of 10, it looked like I was mixing up strawberry milkshakes in the lab. And while I was staring at the fermenter and we were feeding this methylotrope methylene chloride from a vapor phase, what happened is a phage got into it, and I literally watched an OD of 10 go to an OD of 0 right before my eyes.
Wow. So it was a lytic phage.
A lytic phage came in and lysed it. And of course,
well, instead of strawberry milkshake, it was strawberry soda.
It was devastating. And so you can understand if you're and these cheese factories use an incredible amount of milk. And these authors elected to undertake an analysis of phage-host interactions in order to understand how problematic phages recognize and infect their host bacterium. And as we were talking before we started this podcast, Vincent brought up that's effectively how CRISPRs were discovered. It was because of a phage infestation, and the microbes that were resistant to the phage actually had CRISPRs. And so what we can learn from looking at phages and other things is really pretty clever. So the paper is straightforward, but it required a significant amount of work on behalf of the authors. It's organized into 4 parts. Part 1, they describe how they isolate the phages. And this is something that you might want to do if you're looking at the microbiome or something else. So they give you a method, and the method is relatively straightforward. We'll get to that in a second. Then they looked at the genome organization. And again, you renew your understanding of the elegance of viruses and how they do things in a logical format, unlike the bacteria where everything is just sort of thrown in with some rhyme and some reason, but it's harder to figure out. The phage are really good about that. They then talk about tail and tip functions and how they harbor carbohydrate binding domains, that receptor interaction between the virus and the host. And then they talk about morphological analysis. And since we paid tribute to Alan Campbell two podcasts ago, the phage that actually infects this particular streptococcus is actually in the family of COS phages, which COS stands for cohesive ends. And so you have a linear molecule of double-stranded DNA in the phage head. And of course, the sticky ends upon the DNA going into the cell become circularized. And of course, it can integrate or it can go lytic. And so what this work entailed is they took whey samples. And whey is a byproduct of the cheese manufacturing process. And it's the liquid remaining after the milk has been initially curdled and strained. And that's where the phages effectively are. And what this manufacturing plant does, in addition to making cheese, is they make whey protein. And the way you make whey protein is you effectively spray dry it, which means you take the liquid and you put it in the equivalent of a pressure washer and you blow it out. And then the decompression, as the liquid comes out at the high pressure nozzle, it just falls out. So the phage are effectively given a tremendous amount of kinetic energy to remain aloft in the atmosphere of the cheese manufacturing plant. And that's how they get into the process of making the cheese. And the cheese is the classic Irish hard cheese or the Dubliner cheese from the County Cork. And that's where effectively the author's from. And it's made from cow's milk, and it's aged over a year, and it has the hard texture similar to cheddar. So starting in 2006, they isolated a single phage. And they did this via the classic double Agar Plaque assay. And I'll put into the show notes the reference to the method that they used and commenting on how they did it, along with an illustration about how the double Agar Plaque assay is used. And they have a couple of tricks that you need to know about when isolating phage from this particular group. You have to help things along by putting calcium chloride into the plating medium in the soft agar, and the plates have to be dry and they have to be the right nutrients. And so they were able to effectively get a number of phages out of the system. And in all cases, they use the same master stock of host, or ST, and it was the parent strain. And they continuously obtained this from the manufacturer of this inoculum so that it was always the same source of the host. So they knew what they got was indeed appropriate. They also then went out and they got whey samples from other plants. And the second approach that they used to isolate phage involved 2,043 whey samples over this 10-year period. And they tested them against a panel of 52 variants of ST. And they asked the question, did any of the whey samples plaque on these particular strains? And they effectively report out that they did effectively 106,000 characterizations. And so they got a bunch of phage out of the system. And one of their most interesting findings is that throughout the testing period of a decade, despite the fact that the strain is only used for at most a third of the year, namely the parent host strain, ST1, that the phage were present throughout the year. So where are they coming from? And the answer is the whey protein production, which is present in the plant, is likely the source of the phage. And the phage isolates with minor modifications were characterized by restriction analysis. And then if they differed a little, they further test them by multiplex PCR. And then if they were really different, genome sequencing. They show a beautiful series of figures in which they were able to illustrate 17 genetically distinct phages, 8 of which were isolated from the factory from cheese whey samples in 2006, 2008, and 2015. And between 2008 and 2015, they got over 100 phage isolates that were nearly identical to the first phage that they isolated back in 2006.
Can I interrupt you for a second? What's the
Sure.
take-home lesson? Yeah. What have we learned?
Is that the inoculum starter culture is pretty stable in terms of its phage predilection, and that the phage effectively are adapted to this particular dairy plant, that not these peculiar phages aren't able to get a foothold into the factory. They continue to isolate ST1.
But isn't this kind of predictable? Isn't this what you'd predict?
You would predict, yes, indeed. But what they then did, and this is the last aspect of their paper, in order to assess the extent of phage diversity that were acquired from these other factories, because they're trying to understand if an aberrant one gets in and should it be a better infector or it will change the flavor profile. They wanted to really understand it. And so they then determined the phage lineages, a la what we see with a lot of different species going on in the microbiome. And they only found 4 different phage lineages. 2 were very similar to what they found in the factory, and the other 2 lineages were distinct. And as you say, what's news here? And to greatly distill the results from the genome organization of their manuscript is that they learn from a focus analysis of the genomic region that encodes the host recognition functions, which is the tail tip. This highlights that the associated proteins harbor a variety of carbohydrate binding domains, which then corroborates your notion that the phages of this Streptococcus thermophilus recognize carbohydrate receptors at the initial stages of the phage cycle. And these carbohydrate receptors don't seem to change all that much, hence the reason I think we don't see much of evolution of going on. But
so there's a strong selective pressure for maintaining that carbohydrate binding, and any variance would just be lost from the population. Is that
correct?
How we should think about it? Okay.
That's how you should think about it. And, that's basically the paper in a nutshell.
Michael, they're not trying to make resistant bacteria at all, right?
They're just asking who's there.
Is that something they might want to do? Because, as you mentioned, the CRISPR originated from a yogurt plant trying to identify bacteria resistant to phages because the phages were causing problems in the production. And that's where they discovered CRISPR. So couldn't they do a similar thing? Or maybe not. It's not an issue in their production.
So a host mutant? You're thinking about a host mutant?
Yeah. I mean, you could, the simplest thing you could do is simply remove the carbohydrate.
The tail and tip function of the phage protein, which harbors the carbohydrate binding domain, is the important business end.
Yeah, so you could remove the carbohydrate from the host. Or you could just put a CRISPR array in that is, known to inhibit this phage if they wanted to do that.
Yeah, you can certainly try to make bugs resistant to the phage, but the phage is going to mutate again.
Maybe.
Pretty soon you have a warfare that's no end.
Maybe.
Yeah, and so they just wanted to know what was actually going on. And, there were principally, 3 remarkable events that they talked about. First is that phages predominate in dairy samples where the Streptococcus is the predominant starter culture. Second, phages were abundant in whey protein powder. Third, these ST phages are synonymous with modular exchange in their evolutionary developmental pathways. And I didn't get into the nitty gritty of looking at all these open reading frames.
Thank God.
I'm sorry. I did that for you.
I don't know what the take-home lesson is, other than this is a practical study about something that's very important to their industry. But I don't know that I learned anything I didn't know before.
Well, this is a major commercial industry, and in the end, longitudinal studies such as this one are important to understand the natural evolutionary processes that are at play in this industrial context. And
Elio, what I learned is that bodybuilders use whey, and they probably are eating a lot of phages.
They are indeed eating a lot of phages.
That's right.
So, all right. I'm sorry it wasn't as exciting as it should be, but it's a good object lesson of looking at some of the methods and some of the other aspects.
Yeah,
I like the title that has Irish dairy plant in it. I think that's pretty cool.
Nice.
The Irish dairy plant, and you can get this cheese. I spotted this paper a few weeks ago, and I was in Costco this past weekend, and there the cheese was sitting in the Costco dairy case. So I was ready to pick up a chunk.
Yeah. I was wondering, and I didn't pull it out of the methods, whether every time they sampled, did they just pick one plaque of phage, or did they choose 10 and make sure that at any one time point
it wasn't
there was only one type?
It wasn't
in the population.
delineated in the method itself, and I didn't go to see if there were supplemental methods.
Because in Legionella surveillance methods, it's becoming clear that there's typically heterogeneity in any population. So if you're satisfied with just one bacterium, and then the next time you sample, you pick another, you may not realize that there's variety, and you've got to pick, 10, so that you sample the whole spectrum each time.
Yeah,
and then you can begin to understand what's evolution and what's just sampling error, essentially. But,
so that's it in a nutshell. Phage are everywhere, and whey protein is loaded.
The way to make whey.
The way to make whey.
Okay, thank you, Michael. I want to tell you about the upcoming meeting called ASM Microbe, the annual meeting of the American Society for Microbiology. This year it's in Atlanta, Georgia, And ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, which is June 7th through 11th in Atlanta, using the promo code ASMPOD. Delve into your scientific niche in 8 different tracks. Visit asm.org/microbe. That's asm.org/microbe, and use the promo code ASMPOD. It's all one word, for $50 off registration. When you go to register, you will see, do you have a promo code? You type in ASMPOD. All right, now from Elio, we have a paper. What do you have, Elio?
All right, I'll read you the title, which appeared. It's a paper that appeared in MBio, a journal that's thriving, by the way. The title is Physiological Heterogeneity Triggers Sibling Conflict Mediated by the Type 6 Secretion System in an Aggregative Multicellular Bacterium. That's not a mouthful. And the authors are Vera Trojel, Anke Treuner-Lange, Lotte Sjögard-Andersen, and Daniel Wall. I know Daniel. He is at the University of Wisconsin and does some amazing work with the subject of the paper, which is
Wyoming.
Mixo.
It's actually Wyoming. The other
an
W.
aggregative multicellular bacterium. So the paper is quite remarkable in that there is some very nice experimental work and shows something novel, but it's also geared towards understanding the whole idea of multicellularity in microbiology. So the paper has a beautiful combination of experimental work and some philosophy. And I must say, I liked it very much. I enjoyed this paper. So since we have a few minutes, I have to go back and talk a little bit about differentiation and multicellularity in bacteria, a subject that does not occupy the minds of these traditional E. coli-oriented people that I used to represent, I used to belong to. E. coli is not supposed to be a multicellular organism, and since E. coli is the paradigm for everything, or used to be, multicellularity became a subject sort of on the side. However, in one sense, multicellularity in the microbial world is all over the place, and increasingly we have the sense that most bacteria in the universe are sessile. They sit on a surface. They either make biofilms or something else, which represents a different idea than bacteria growing in L broth in a flask and being all by themselves. So multicellularity in bacteria can mean something as, if you wish, simple as making a fairly nondescript biofilm. Although biofilms are complicated and heterogeneous, it's still a fairly simple sort of a thing. Contrast this with what happens in the world of eukaryotes, where, I'm looking out my window and I see palm trees, and I see a dog walking around, and bacteria never developed into something as complicated as that, right? They stayed relatively simple. So, but within the bacteria, there are some which are capable of significant differentiation, and these are the Mixo Bacteria. Mixo means slime. So Mixobacteria are something akin to Mixomycetes, that is slime molds. They're not, of course.
And they're modal by gliding motility.
That's right. They're mortal. And let me say something about that. The perhaps best studied, or one of the best studied of the Mixococcus is a bug called Mixococcus Xanthus, which means yellow. And this has been studied now for quite a few years in some wonderful laboratories that have added a tremendous amount to our knowledge. Mixococcus Xanthus is capable of 2 kinds of differentiation, not just one. One of them is in its motility. It has 2 kinds of motility. One is fairly straightforward. It's where single cells follow a trail of slime. This is called adventurous motility. And contrast this with what is called social motility, which is when cells get together in what is called a raft or a swarm and move along the surface doing that. So why are they doing it? So there are 2 kinds of motilities. One is simple, the other is differentiated. So they do it because they're looking for prey, and the prey is other bacteria. They eat other bacteria. So when a single cell or a swarm of cells finds a set of bacteria, they gobble them up. The swarms, by the way, are also known as a wolf pack because they act like that. Okay. So this is kind of fascinating on its own right. Okay. By the way, the way they eat other bacteria is by inducing them to lyse, by secreting hydrolyses that destroy the bacteria, and they use the food that's so developed. This is a very cooperative sort of a thing because the secretion of the metabolites by the dead bacteria is now used by the whole swarm. So it's really quite interesting. But when food is scarce and they can't get any food by swarming around, what they do is they aggregate into what amounts to fruiting bodies. Fruiting bodies is a term that is used in mushrooms. A mushroom is a fruiting body of a fungus. These are not fungi, but the term fruiting body is appropriate because they stick out from the surface. And these are seen on dung, in soil particles, all over the place. They're not uncommon at all. The fruiting bodies of the Mixococci can be very simple, like in Mixococcus Xanthus. It's really just a hill. Looks like a little hill of cells, or quite complicated, in which case it looks like beautiful forms, shapes, which branch with branches and so forth. I wish, well, we'll put this in the show notes to
kind
show
of like
people
mushrooms.
Mm-hmm.
Sorry?
I mean, beautiful structures of mushrooms. They can.
That's right, mushroom-like things or fungus-like. Anyhow, so this is social motility, and it is a form of multicellularity. Remember, the title said the study is in aggregative multicellular bacterium. So aggregative means here that it makes an aggregate of like cells. Now, here's a bit of a story. When we think about multicellularity, let's say in higher forms of life, or complicated forms of life, we start out usually with an egg that was fertilized, and that's a single cell that becomes 2 cells, becomes 4 cells, etc. All of them identical until something unusual and unlikely happens, namely a somatic mutation. So by and large, when you contemplate at animals and plants, they are all clones of a single cell, and that is required for their aggregation ability. That is, if you want to aggregate into a structure like a stem of a plant or an organ of an animal, you better have cells which are alike. So that is solved in multicellular organisms by starting with an egg. This is not solved in bacteria because the aggregation requires the coming together of potentially different cells, unlike cells. And if you did that, you would not have a very successful aggregate, because if the cells are distinct, they each are going to pull their own way. Okay, they're not going to say, let's all pull in the same direction. Some are going to pull this way, some are going to pull the other way. So this is a problem, and that's the problem that is addressed in this paper. So I would say that generally speaking, one can learn about differentiation from studying bacteria, even though higher form of differentiations are not common. They do exist. By the way, as an aside, these bugs that are capable of this kind of differentiation, are notable for having the largest genomes in the microbial world. There's one guy called Minicystis rosea, which has a genome of 16 million bases.
Wow.
Okay, that's about better than half of what we have in terms of genes. So this is why do they all have large genomes?
Sorry, Elio, what was that number again?
16 million.
Million bases.
16 million. Well,
that's 4 times the size of E. coli.
E. coli, right? Okay.
Right. And this is the world record in terms of genome size among microbes, among bacteria. I hope you're impressed.
I am.
Well,
Suggest they're very versatile eaters, and then this multicellularity must take a lot of their genome too.
Elio, I'm not impressed because our genome is 3 billion bases,
Yeah, that's the implication. If you want to make something complicated, you better use a lot of genes. Well, the meaning of that we'll let go for the time being. So let's.
But let me just say one thing, Elio.
Sure.
The genome of some flower, I can't remember it now, is huge, bigger than ours, bigger than the human genome. So size doesn't always mean complexity. There's some other things involved that we don't understand. Just wanted to say.
You're right.
Okay.
But we're talking about bacteria here, so we're impressed. Yeah, there's some plants that have an extravagant number of genes. So
yes, exactly.
Anyhow, so what's the problem here? The problem here is how do these cells recognize their kin? Okay, and how do they avoid making aggregates with non-kin? So the experimental setup that they use is very simple. They take the strain of M. xanthus and mutagenizes to make auxotrophs, that is, mutants which are not capable of making certain amino acids, and they cannot grow unless you supply them with amino acids. So they have two strains, the wild type, which is capable of growing on minimal media, doesn't require amino acids, and a mutant, which requires certain given amino acids.
That was a beautifully elegant strategy, wasn't it?
Auxotrophs always are.
Yeah, I just thought that was a lovely strategy to control the growth.
Okay. So I want to do another little bit of philosophizing, because they have this lab, the lab of Wall has found another thing which is quite unique among the Mixo Bacteria, and that is that when the way they recognize each other is by something called outer membrane exchange, which consists of cells binding to each other to effect a give and take of outer membrane proteins. Remember, these are gram-negative, so they have an outer membrane, right? So this is a way of communicating between bacteria by exchanging outer membrane proteins. I hope you agree that this is interesting. And it serves either good things like it can help damaged cells recover, or not so good things like delivering toxins to neighbors. But this is an aside. I'm sorry. I'm just to show you that these are interesting bugs. I'm plugging Mixococcus here. Okay. Anyhow, but what we discuss here is something different. The recognition of non-self is carried out via a different method than the exchange of outer membranes. It's called type 6 secretion. So what's type 6 secretion? Well, it's obviously one of many types of secretion, which we have discussed numerous times here. And it was discovered in Macelano's lab, I believe, in Vibrio cholera, where he found that strains of Vibrio cholera will kill other bacteria, and they do it by having an injection apparatus, which is characteristic of several secretion systems, not just this one, but this is separate and different. And here the apparatus pierces a target cell to deliver toxins or other cargo. Okay, so here this is where the conversation takes place between bacteria by the type 6 secretion systems. So the authors examined the interaction between genetic siblings that differ physiologically, namely the wild type and the auxotroph. So when you put this in minimal medium, the wild type will grow, and the parent strain will grow, and the auxotroph will not. So let me quote what they say. They say, we ask whether this kin cell antagonize, cooperate with, or act differently towards one another. We found that antagonism occurred between kin M. xanthus strains only under conditions under which they were physiologically distinct. In other words, kin likes kin, okay? Nothing happens if you're just one kind. But if you mix the two kinds, as they say, we show that this antagonism is depending on the type 6 secretion system. And they say we identify the effector immunity pair that mediates this interaction. Let me stay with it for a second. If you are going to produce a toxin, you have to do something else. You have to become immune to the toxin. Otherwise you keel over and die. So when the effector is injected, it will probably inject both a toxin and an antitoxin. This reminds you of the subject of programmed cell death in bacteria.
Mm-hmm.
Well, it should. Okay, because the system there of toxin, antitoxin, just to remind you, requires that the toxin be neutralized by an antitoxin, but the toxin turns to be stable, the antitoxin unstable, which has enormous consequences. So how did they arrive at the conclusion that kin likes kin and does not and destroys the non-kin? That is, the wild-type cells, which are capable of growing in minimal medium, will kill the non-growing. Let me remind you that just being put non-growing does not mean you're going to die right away. So an auxotroph put in a minimal medium will not die to a significant extent for a while, even though it can't grow. But if it's put together with the wild type will inject its bad stuff into it and die. So they show by nice experiments that this is a dependent type 6 secretion system, and as expected, M. xanthus carries a gene cluster for the type 6 secretion consisting of 13 core genes, and this is required for both killing and immunity. So what is it that it transfers? The putative toxic material is called TSXE. Turns out to consist of proteins involved in peptidoglycan degradation and other hydrolytic activities. In other words, the toxic material will destroy a recipient cell unless it becomes immune. So wait a minute, why isn't the auxotroph protected by the immune component that it's supposed to be carrying? Okay, so the reason the authors pose it is that all these experiments are eventually done under conditions of starvation, which is where if you remember what I said earlier, the fluting body formation requires starvation. So under these conditions, there's a reduction in the synthesis of the toxic and antitoxic material. And since the material involved in immunity is likely to be unstable, the level decreases to where protein synthesis is inhibited. So when you starve and the wild-type cells inject their toxin, antitoxin, the recipient cells, the mutants, will die because eventually the toxin remains stable, but no new toxin, antitoxin is made, and this is the way they see it. So, you with me?
Yep, got it.
Yep.
So the paper I think it's really an amazing paper because it's rich in interesting theoretical conjectures. They let themselves go with a lot of thought, and I like that. They point out that heterogeneity in a population can often be beneficial. For instance, bacterial cultures can carry dormant, persistent cells that are resistant to antibiotic. So they're heterogeneous with regard to sensitivity to antibiotic. And so some are resistant, which is good for business. However, in cases where the population fitness depends on cooperative and synchronized cell behaviors, such as for social or aggregative multicellular organisms, the ones we're talking about here, where here you want cooperation, the population heterogeneity is a challenge that hinders the ability to coordinate behaviors. In other words, if you have cells which are different, you are not going to be able to make a fluting body, for instance, or you may not be able to make a raft.
So they wipe out the stragglers.
They're separating the wheat from the chaff.
Yeah, that's it.
That's right.
So the way to handle this, in this case, is simply to kill one of the guys.
Mm-hmm.
And a similar situation is found in Bacillus subtilis biofilms, where cells make a matrix in order to make a biofilm, and cells that do not produce a matrix are cannibalized by the matrix producing siblings. So this is kind of the story. Now the mechanism. Let me say that the mechanism of death is not clear, and the mechanism of immunity is not clear. They need to really go in and study that. So right now we're on hold with regard to this. But the conclusion is that multicellularity offers many advantages, Obviously, if you're bigger, multicellular, you're going to be bigger, and you can avoid predation by relatively small predators. You can also exploit new environmental niches and develop specialized functions that allow complex and powerful behaviors. In bacteria, this doesn't happen all that much, but when it happens, and one doesn't know why. This is really a general question. Why is it that bacteria did not learn how to become trees, dogs or cats?
Maybe they did.
We don't know.
But
maybe they did. But no.
But dogs and cats and trees have, dogs and cats have bacteria inside of them.
They do, and they're part of it, but they're not.
Yeah.
You can't say a bacterium became a tree.
No. Well, they don't make enough energy to make those multicellular.
But that's a popular view that the reason developmental development happened in the eukaryotes because they have mitochondria.
Exactly.
That is, they can make a lot of energy. Yeah. That's a view that's held
also
by
oxygen. Remember, oxygen is relatively new to the planet, and the bacteria have been bound much longer than there's been oxygen.
Yeah,
sure.
But they couldn't use it as efficiently as a mitochondria can, right? And that's the difference.
Well, anyhow,
Elio, let me ask you a question. So this is done using auxotrophs, right? How do we know that this is done using auxotrophs, right?
Mm-hmm.
Right.
How do we know that in the real world this happens? Maybe there's something weird about an auxotroph that stimulates this killing. How do we know that this sibling killing would happen in nature where these bacteria are
That's
living?
a very good question. That's a fine question. And I think that it's possible to study this, but in nature, in a way, an agar plate is nature also.
Well, I'll volunteer that in pathogenesis, a lot of the pathogens that attack humans are auxotrophs. They lack the ability to make a particular amino acid or a vitamin or some other growth factor. And so auxotrophs are quite common as pathogens simply because the host is supplying them. And in the case of these organisms that are growing growing in the environment, auxotrophy is a normal behavior because the thing that the microbe is interacting with, namely they're eating other bacteria, their other bacteria are going to supply them with the necessary growth factor or the necessary amino acid that they may not be able to make, and that will give them the selective advantage. They don't need to invest the energy into making something complicated and big.
Yeah.
But they also want to take care of cheaters too, eliminate cheaters that aren't carrying their weight. And so I always thought of Mixococcus as this very cooperative population of cells, but boy, they're ruthless. If you're growing slowly, or if you don't have enough, if you're not as fit as the rest of us, boom, we're killing you with a toxin.
You can give this paper to the graduate students who are not working hard enough and ask them to comment on it in the club.
It's full of food for thought, shall we say, right? Yeah, very good. So I want to finish by telling you a good title for the show would be what my colleague in the blog, Christopher Weigel, did. He termed the strategy of the wild type killing the mutants, kill the loser, as opposed to kill the winner.
Kill the laggards. Yeah, so this work was led by a PhD student in Dan Wall's lab, Vera Trojel, and she describes her path to science as somewhat unconventional. She earned a bachelor's degree in veterinary medicine at the University of Belgrade in Serbia, where she grew up, and then worked as a veterinarian for several years. But then she decided to pursue her interest in research, and she applied to the Molecular and Cellular Life Sciences program at the University of Wyoming and is now a graduate student there. She's remembering that during her first year, she was able to rotate or sample 4 different labs: cell biology, cancer biology, microfluidics, and molecular biology. And through that process, she realized that microbiology was her primary interest and also her passion. She's fascinated by the complexity of microbes, especially by the intricate mechanisms and tools they use to communicate, cooperate, and compete to survive in the ever-changing environment. So as we saw here, she's focused on how bacteria interactions are influenced by genetic and physiological heterogeneity. And she wants to continue this line of investigation and is considering possibly looking at microbiome and human health and disease or host pathogen interactions. She's grateful for the friendly, supportive, and stimulating environment that she's had with Daniel Wall's group there at the University of Wyoming. And she does have some advice for junior colleagues. She says, never lose your scientific curiosity or sense of wonder and joy of discovery, because it's those emotions that will carry you through and provide resilience through the unavoidable setbacks and challenges that experimental research has. She also said that being an international student has had its own challenges. She's away from her family, her friends, her home. And she's found the best way to cope with that is to continue to cherish meaningful relationships and contacts with people you love, and to invest the effort to build new friendships in her new temporary home. Outside the lab, Vera enjoys art, books, and fitness. And although she's no longer working as a veterinarian, she still devotes some free time to helping animals by volunteering in the Laramie Animal Shelter and also in one of Colorado's farm animal sanctuaries. So that's our first author, Vera Trojel, who has led this work, and this is her third publication in this general area.
You said Colorado? I thought she was in Wyoming.
She's in Wyoming, but she said Colorado Farm Sanctuary.
Right.
So,
it's the name of it. It's not actually in Colorado then.
Oh.
Maybe maybe they're really close. I don't know my geography, I guess. Let me just check. Maybe it's a half hour away.
Make the Four Corners. Those make the Four Corners. Pretty sure.
I'm checking
it's
it.
West of the Hudson.
West of the Hudson? What's west?
What's west? New Jersey is west.
Oh, that's right. Yes.
You see it out of your window every day.
I do. So here's Wyoming. No, Four Corners doesn't include Wyoming. Four Corners are Utah, Colorado, Arizona, and New Mexico.
Ah, my bad.
So Wyoming, however, is north of Colorado. So she's in Laramie. It's right near the border.
Yeah.
So she could drive right over and find the Colorado thingy you just said.
Apparently she does.
There you go.
And Laramie is also home to the USDA arthropod-borne animal disease research laboratory for the USDA.
Oh,
okay. I haven't been there. My knowledge of the West is lacking clearly, but I do know Michigan. I know where that is. And there's something to do with the palm of a hand that I can't remember.
A mitten.
A mitten?
A mitten.
All right. Thank you, Michele.
You're welcome.
Very nice to hear some personal stories from our authors. Are you done, Elio?
I am done.
Thank you very much.
My pleasure.
That's it for TWiM 177. The next one, 178, will be in Atlanta, so hopefully you can join us there. If not, you can listen afterwards. And how do you listen? Well, you could go to asm.org/TWiM, or you can listen on your mobile device like a phone or a tablet, in which case you probably use some kind of an app to play the podcast, and you can just search for TWiM. Now, you might find other podcasts with the same name, so make sure you're getting This Week in Microbiology. Subscribe. It's free, and you'll get every episode automatically. We post about twice a month, so it won't fill up your phone. So please do that. And also consider becoming a patron of TWiM and all the other science shows that we do. You can go to microbe.tv/contribute. We have a number of ways that you can help us out, including you could give us, say, $1 a month, and that would really help. If every listener did that, boy, we could do all kinds of things. And, of course, questions and comments, twim@microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure. Thank you.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Again, thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I'm Vincent Racaniello. You can find me at virology.ws. And we will all be in Atlanta for ASM Microbe next, as I mentioned. And I want to thank ASM for their support of TWiM. Ray Ortega for his technical help. If you want to see Ray, he'll be at ASM Microbe. And I want to thank Ronald Jenkins for the music here on TWiM. Thanks for listening, everyone. See you next time on This Week in Microbiology.